In the first slide, you can see impaired innate immunity in fruit fly, the mutation in the toll pathway. In 1980s, some German scientists discovered that a protein called toll is important for the dorsal ventral axis determination of Drosophila. The toll word is actually a German slang. It means weird. Those Drosophila have mutation, have a non-functional toll protein. They have a visually scrambled anatomy. But subsequently, scientists also discovered that those Drosophila have this mutation in toll protein that actually determine its dorsal ventral axis. They are also highly susceptible to a infection by a fungus called Aspergillus fumigatus. So in this picture, you can see a Drosophila is actually infected with fungus called Aspergillus fumigatus. So now I want to introduce you the structure of toll-like receptor. So in mammals, it is called toll-like receptor. The toll-like receptor contains two major domains. One is called leucine-rich repeats. It is the external domain, and a cytoplasmic domain called TLR domain that is actually responsible for the cell signaling. So in this case, I want to mention that toll-like receptor cannot work as a monomer. It always forms a dimer. It can be heterodimer or homodimer. Majority of the toll-like receptor function as a homodimer, but 3 molecules of toll-like receptor are function as a heterodimer, like TLR1, TLR2 and TLR6. This TLR1 and TLR2 form a heterodimer, and TLR2 and TLR6 form heterodimer. Rest of the TLR toll-like receptor protein form homodimer. There are 9 toll-like receptor. So you can see here the toll-like receptor 2 and 1 bind and form a heterodimer so that it can bind with a lipopeptide, and the TLR3 and 3 form homodimer that can bind double-strand RNA. Toll-like receptor are under the pattern recognition receptor. So this can identify PAMP, means pathogen associated molecular pattern. You can see here that toll-like receptor 2 and 1, if it is monomer in nature, it cannot bind with a lipopeptide. After forming the dimer, it can bind with the lipopeptide. Toll-like receptor, you have to just remember 2 important points. One is the function of each toll-like receptor and their position. So the cellular location of toll-like receptor are different. One set of toll-like receptor present on the plasma membrane and one set of toll-like receptor present on the endosome. So you have to remember, like TLR1 and 2 are present on the membrane, plasma membrane, and 4, 5 or 6 are present on the plasma membrane. So total TLR1, 2, 4, 5 and 6 are present on the plasma membrane, and TLR3, 7, 8 and 9 are present on the into the endosome. In this slide, it is also showing the function of each toll-like receptor. In my previous slide, I have mentioned that toll-like receptor cannot function as a monomer. You don't think that the TLR5 acts like a monomer or TLR4. Actually, it is not showing the dimer, but TLR5 actually works as a dimer. So TLR5 and another TLR5 form a dimer, can bind with flagellin. So from the beginning, TLR1 and TLR2 can bind with triacyl lipopeptide. These are present on the bacteria, and TLR2 and TLR6 can bind with diacyl lipopeptide. Now TLR4 can bind with very important components of bacteria called lipopolysaccharide. It is also known as endotoxin. It present in gram-negative bacteria. So toll-like receptor 4, TLR4, form a homodimer, TLR4 and TLR4, that can bind LPS, recognize LPS. LPS is one example of PAM. TLR5 can bind with a 5 and form a homodimer, can bind with flagellin. In the endosome, the TLR3 can bind with double-strand RNA. TLR7 can bind with single-strand RNA of virus. TLR8 also form homodimer. So they all form homodimer. TLR3 recognize double-strand RNA. TLR7 and TLR8 recognize ssRNA. TLR9 can recognize CpG DNA. Our immune system has a toll-like receptor that can recognize CpG molecules because bacterial CpG molecule and viral CpG molecule differ from eukaryotic CpG molecules. In eukaryotes, majority of the CpG molecules are in methylated forms. Approximately 70% of human CpG molecules are methylated, and in case of bacteria and virus, most of the or all CpG molecules are unmethylated in condition. So the toll-like receptor can recognize unmethylated CpG DNA. So toll-like receptor can recognize PAMP. And the main important point I have mentioned that the toll-like receptor cannot function as a monomer. It always forms a dimer. It may be homodimer or heterodimer. Majority is homodimer and minority is heterodimer like TLR 2.1 and TLR 2.6. And after binding with the PAMP or pathogen associated molecular pattern, toll-like receptor starts a signaling procedure or signaling cascade. Don't have to remember all the signaling molecules. Just look at the last molecules that are transcription factor like NF-kappa beta. After activation of a toll-like receptor or the toll-like receptor when bind with PAMP, it actually produce NF-kappa beta. Another important transcription factor is AP1, and the last two important transcription factor called IRF7 and IRF3. So these 3 or 4 transcription factor actually sends signal, as because they are transcription factor help in translation to produce some proteins like NF-kappa beta, AP1 or IRF3 or 7 helps to produce some antimicrobials from the cell who have recognized PAMP, some cytokines and chemokines. And another important chemical is called IFN alpha and beta. This is called type 1 interferon. This type 1 interferon is actually responsible for immunity against viral. So this type 1 interferon develop antiviral immunity.